What we showed was if you take this progeroid mouse that ages 6 times faster than a wild type mouse because it has more DNA damage, is missing the DNA repair complex. So it acquires more damage, acquires more senescence, ages faster. And we showed their stem cells in these mice are not functional. If you damage the muscle doesn't repair itself. If you isolate stem cells, they don't grow very well in culture. But we showed if you put young, healthy stem cells from a normal mouse, not the progeria mouse, into this model, that we could rescue partially lifespan and extend health span. And this was only 6 million, 5 million stem cells injected into this mouse, where every cell in that mouse has more DNA damage and showing signs of senescence. 6 million stem cells was able to partially rescue that phenotype. So this was very exciting because it means that stem cell dysfunction is not only a marker of aging, it actually contributes to aging. Because if you now put back young functional stem cells, it restores many of the youthfulness of that mouse model. And so that led to us trying to understand the mechanism for how these young stem cells could extend health span and lifespan in this model of accelerated aging.
Hello, Dr. Robbins. You are a professor of biochemistry, molecular biology, and biophysics at the University of Minnesota, where you run a lab studying the biology of aging, including senescence and stem cells and exosomes. So welcome back to Modern Healthspan, and thank you so much for joining us today.
Thank you for having me, Richard.
So, Dr. Robbins, I believe you are investigating kind of the impact of factors from stem cells on senescent cells and maybe other markers of aging. What I would like to start with, though, is an understanding of the role of stem cells in aging. How do they change as we get older, and how does this impact us? So maybe, how do stem cells change with age?
Right, it's a great question, and it's something I think we're still trying to sort out. So everything that we've done has actually been looking at the function of stem cells. And the function we have, there are really two general types of ways of measuring that function. One is you can create damage in the tissue, and we use animal models, but this could be an injury you have while running in the muscle. And as you age, the ability of the stem cells to repair that damage are reduced. So the amount of repair of that damaged tissue is less. And you can see that in a muscle injury, there's more fibrosis, less muscle fibers after injury in an older animal than there is in the younger. The second way that we look at this is we actually isolate the stem cells. So we can do this from bone marrow, we can do this from tissues, we can grow them in culture, and then can test their ability to differentiate into bone, muscle, cartilage, etc. And we show that if you isolate stem cells from older animals or from older humans, they don't differentiate into the different lineages as efficiently. And if you put them back into an animal, they don't work as efficiently to repair damaged tissue. So they're functional assays to show that this activity of adult stem cells is reduced with aging.
It's the actual stem cell that's becoming less functional rather than the niche that it's coming from, or is it both?
It's a great question, and I think the answer is it's both. There's evidence that if you target other so-called hallmarks of aging, such as senescence, the epigenetic profile, improved proteostasis, et cetera, that actually leads to improvement in stem cell function. And so one example is if you reduce the senescent cell burden in older animals, their stem cells become more functional and they can repair tissue more effectively. And so that's probably an indirect effect because you're getting rid of the inflammation caused by senescent cells that leads to stem cell dysfunction. And so there's probably both effects on the stem cell as we age and effects on the niche, which may be driven by inflammatory factors, other secreted factors that then indirectly affect stem cell function. So we would like, if possible, to both rejuvenate stem cells in the older individuals as well as the niche for those stem cells.
Right. So one of the hallmarks of aging is stem cell exhaustion, that,
Right.
which I never really fully understood. That means that they don't function well, or we're running out of stem cells, or does it mean both?
Well, so the thought is, and unfortunately, it's hard to measure stem cells, the number and their function in vivo. You know, we can damage a tissue and look for repair, but it doesn't tell you anything about numbers, and the overall function of all those. Um, and so when when we're thinking about what happens to the stem cell in vivo when it becomes what you say exhausted, is it really exhausted, or is it losing function, or are we losing numbers? And I don't think we really know for all the different types of adult stem cells. What we measure is really loss of function. And we quantitate stem cells by markers they have on their surface. So we can actually measure the cells that have certain markers that we say are present on stem cells. But that doesn't truly make them a functional stem cell or exhausted stem cell. So the field is still, I think, not addressing this accurately because we don't have the right tools to measure stem cell function in vivo with age. It requires taking cells out of the animal, expanding them in culture, characterizing them in culture, and putting them back in. And then they've been manipulated in ways that aren't necessarily physiological.
Okay, so so exhaustion is almost like a metaphor. I mean, it's
Yes.
like they're exhausted rather than actually.
And in that term, actually, it's applied to the immune system extensively. So we have terms called immune exhaustion that our T cells, after fighting off infections for long periods of time, become dysfunctional. We call them exhausted. But it appears that you can actually restore their function. So they may be exhaustive, but you can actually tweak them in certain ways to make them more active. And I think the same is with stem cells. So I think it's just a term that may not necessarily be an accurate term. It's
Okay,
more of a reduction in function. And I think there are ways we could tweak that to try to restore that function in those dysfunctional stem cells.
so one other question in this area. So there are senescent stem cells. So what is, I guess, what makes a senescent stem cell, and what is a senescent stem cell? Because I thought like a senescent cell was one that can't replicate, but a stem cell is one that can reproduce, that can replicate. So it seems almost oxymoronic.
Well, I would say, and there are probably papers that say they're truly senescent stem cells in vivo, but I don't think we really know if they're truly senescent. There are not many stem cells. We haven't really characterized them carefully in vivo for markers of stemness and markers of senescence. So there may be a senescent-like state. Where we've seen senescence in aged stem cells is if you actually isolate them from different tissues, again, bone marrow, muscle, adipose tissue. And if you put them in culture, they very quickly show markers of senescence as compared to stem cells from much younger animals. So they senesce faster in culture upon stress. But if that's actually occurring in vivo, we hypothesize it is. But I have to say, I don't think there's any really solid evidence to show they're truly senescent. And again, there may be senescent cells in the niche, which then affect the stem cell function. And whether the stem cells are truly the senescent cells, I think is still poorly defined.
Right. So whether the niche, I guess the cells that contain the niche are senescent and releasing the SASP, or the stem cells within it are releasing the SASP, is not
Right,
entirely clear.
right. And I would say it's probably the stem cells are not truly senescent. Um, Senescent cells usually, it reflects amount of damage, many times DNA damage. And the thought is that stem cells actually have better DNA repair mechanisms. So they don't acquire the same amount of damage because they're protected because you want your stem cells to be functional. So I would say it's much harder to make a stem cell senescent than it would be another type of cell in vivo. But there's enough stress, enough damage from older individuals to their stem cells that if you take them out of the body and try to manipulate them, they senesce much faster.
So you were starting to look at mice studies to investigate the impact of young stem cells on older individuals. I think they were progeroid
Right.
mice.
That's correct.
Yeah, so could you talk a little bit about what you found there when you transplanted the young stem cells?
Right. So yeah, so this has been an ongoing project for the last 15 years, but the observation we made, I think around 2010 in collaboration with a group at the University of Pittsburgh, Johnny Heward's lab, Laura Niederhofer's lab, and mine. I was a faculty member there for many years. What we showed was if you take this progeroid mouse that ages 6 times faster than a wild type mouse because it has more DNA damage, is missing the DNA repair complex. So it acquires more damage, acquires more senescence, ages faster. And we showed their stem cells in these mice are not functional. If you damage the muscle doesn't repair itself. If you isolate stem cells, they don't grow very well in culture. But we showed if you put young, healthy stem cells from a normal mouse, not the progeria mouse, into this model, that we could rescue partially lifespan and extend health span. And this was only 6 million, 5 million stem cells injected into this mouse, where every cell in that mouse has more DNA damage and showing signs of senescence. 6 million stem cells was able to partially rescue that phenotype. So this was very exciting because it means that stem cell dysfunction is not only a marker of aging, it actually contributes to aging. Because if you now put back young functional stem cells, it restores many of the youthfulness of that mouse model. And so that led to us trying to understand the mechanism for how these young stem cells could extend health span and lifespan in this model of accelerated aging.
So the stem cells that were doned, is that a word? Doned. Were they also from young progeroid mice, or they were for young wild
They're
type?
from young wild type. So even from young progeroid mice, their stem cells are already showing signs of dysfunction. So this
Okay.
is from young wild type mouse. So they're highly functional stem cells. And the younger the mouse, the better. If you get them from middle-aged mice, they don't work as well. And if you get them from older mice, they don't work hardly at all. Or you have to inject many more stem cells. So you can actually titrate out function of stem cells by the ability to rescue lifespan and health span defects in this progeria mouse model.
And were they? I mean, are they genetically identical apart from the progeroid problem?
Yes,
Yes.
they're
So
from the
that's why
same
they weren't
strain of
rejected.
mice. Yes. Yes, so there's no immune rejection due to foreign cells being injected into these mice. So same genetic background. The only thing they have is a DNA repair complex, which the progeria mice are missing. So their stem cells are functional.
Now we're going to talk about exosomes in a second, but do we know that? So it's one of the things that the stem cells do, right? Is they replicate. So
Right.
it wasn't because they now had stem cells which had DNA repair and they were replicating and fixing the tissue. That was
Right.
not the mechanism.
So that was the first question reviewers of our paper asked is, are these cells contributing to repair of damaged tissue directly? So we labeled these stem cells with a marker so we could stain them blue. There was a blue marker that you could put into them. And we showed that the majority of the cells stayed at the site of injection and didn't migrate to different tissues. So we couldn't find cells in the lung or in the brain or in the muscle, but yet those tissues were now healthier. So there were no stem cells locally that had migrated to that site, but yet we were seeing positive effects of those stem cells despite the fact they were somewhere else in the body. So that led to the hypothesis that these were factors that are being released by these young healthy stem cells that were then migrating to the different tissues and improving the quality of that tissue and extending health span measurements.
Okay, right. And then you did some experiments to see if that was the case, like separating the stem cells from in vitro from the, I guess, from the tissue.
Right. So what we showed, and so we used assays that were available in the laboratory, and so the two assays we used to try to look at what the mechanism could be for this rescue of Healthspan defects in the progeria model was actually culturing young healthy stem cells with progeria stem cells, and showed that in the transwell system, where the cells don't come in contact with each other, but can release soluble factors that can go through a membrane and come in contact with the defective stem cells, it rescued their function. So this showed that young healthy stem cells can release factors that can make an old or progeria-derived stem cell more functional. So that was intriguing. The second thing we showed, and we're doing a lot of work on senescence in the lab, so we have a lot of assays to look for the ability of different agents, whether it's small molecules or biologics, to affect the level of senescence. And we showed again that something that was released from the stem cells, these young healthy stem cells, could actually reduce the level of senescence in other cell types, including stem cells. So we showed we could rescue function of aged stem cells, and we showed that we could reduce the level of senescence and improve the resilience of cells in culture.
Did you know that over 80% of the population is deficient in magnesium, a mineral involved in over 600 biochemical reactions? And there are 7 forms of magnesium, each with distinct physiological targets. That's why I take Magnesium Breakthrough from BiOptimizers. It's the only full-spectrum formula with all 7 forms. Chelate for muscle recovery, citrate for arterial health, bisglycinate for digestion, malate for energy production, sucrosomial for immune function, taurate for cardiovascular health, and orotate for metabolic optimization. Clinical research confirms that organic magnesiums like citrate and malate have higher bioavailability than the common oxide forms found in most supplements. Different forms target specific tissues. Taurate, malate, and citrate show neurological benefits, which is why Magnesium Breakthrough users report dramatic improvements in stress response and sleep quality. If you want to try Magnesium Breakthrough, go to bioptimizers.com/modern and enter promo code MODERN10 to get 10% off any order. And if you subscribe, not only will you get amazing discounts and free gifts, you will make sure your monthly supply is guaranteed. Thank you for your support. Do you know if it was rescuing the senescent cells, or it was suppressing the SASP, or it was killing them?
No, so it's a great question. So there was no cell death. This was not functioning as a senolytic. It was suppressing the SASP, it's suppressing markers that blocked cell replication. It changed the transcriptome in ways that make the cells look a little bit younger, like a younger transcriptome. So many changes, it said that it was suppressing senescence and the senescence-associated secretory phenotype, which are the inflammatory factors that senescent cells secrete.
Okay, so you next I think you look to try and figure out, okay, so what are these soluble factors, and so what did you find there?
Right, so that's we thought at the time it was $1 billion question. It may still be the $1 billion question, but if you had something that young healthy stem cells are releasing that then could affect not so healthy cells at a distance in the mouse or in the human, and we could kind of mimic this in our cell culture assays. Were these single proteins, were these growth factors, were these inflammatory factors, or were these actually larger components such as extracellular vesicles, which is something my lab also works on. So the first thing we do to determine if it's a small protein or if it's a larger vesicle is you separate the vesicles, because you can separate them by centrifugation. They're large enough that you can spin them out of the media from cells. and determine if you got rid of the vesicles, did that media from stem cells or the what's being released, was it able to rescue dysfunctional cells and reduce senescence, or was that activity co-purifying with the exosomes? And when we fractionated what was being released from stem cells, the majority, I can't say all, but the majority of activity co-purified with extracellular vesicles, which gave us immediately a direction to go to start to understand mechanism in more detail.
So one question on exosomes. So there's a number of extracellular vesicles that get released by cells. Whether it's an exosome or something else, is that defined by the size or the contents or some other way?
It's a great question. And as the field is evolving, we realize there are more and more different types of vesicles. It seems like every month there's a new vesicle isolated from a certain cell type of it's defined by the size, the shape, and the content. So we used to say that there were extracellular vesicles that were comprised of exosomes and then larger particles that have a few different names, but they're really two types of vesicles. Now it appears that there are dozens of different types of vesicles, again, defined by a variety of different parameters. And even an exosome, which is derived from the multivesicular body in the cell, and I won't go into the details necessarily about that, but depending where in the cell these vesicles are made, their content can vary. So even from a single cell and a certain size vesicle that's been characterized by certain markers, the content can vary depending what part of the cell that vesicle was originally formed before it was secreted. So vesicles are very heterogeneous in their content, in their size, in their shape, and probably also in their function. So
Okay.
vesicles are really defined as lipid particles that are missing a nucleus. So they're a lipid membrane, very similar to a cellular membrane, but it doesn't have a nucleus. They can be small. They can be 50 to 100 nanometers, or they can be 1 to 2 or 3 microns in size, depending on the type of vesicle and the source.
Okay. And an exosome has a size limit, or how would you define an exosome then?
So an exosome, again, that the definition of an exosome is getting more and more gray as we learn more about them, because it appears they can be made in different ways. But in theory, an exosome they're smaller. They're the smaller type of vesicle, usually 50 to 100 nanometers in size, and characterized by certain protein markers and certain microRNA markers that you can find inside the vesicles. But it appears that exosomes can be generated by release from the cell membrane through budding and from these multivesicular bodies that are produced inside the cell, traffic to the membrane, and then are released outside. So multiple mechanisms for how to generate these exosomes, which now makes it more complicated.
Okay, so then you looked inside the exosomes to try and figure out what were the factors that were making having the beneficial effects, I guess the xenomorphic effects.
Right.
So you said that you chose microRNA as the most likely thing. So could we start with what is microRNA? How does it differ from, say, messenger RNA or any other type of RNA?
So cells have many different types of RNAs. They have messenger RNA that encodes for protein. They have transfer RNAs that are involved in protein synthesis. And then they have a variety of non-coding RNAs, some which are very long, which play certain roles in regulating gene expression. But there are classes of very small RNAs. So microRNAs are one of a number of classes of small RNAs. They're 21 to 23 bases in length. And their role appears to be to suppress or modulate gene expression. So these small RNAs bind to complementary sequences in the messenger RNA that will prevent that messenger RNA from being translated, or it actually targets the messenger RNA for degradation. So depending on the microRNA, depending on the target, depending on the messenger RNA, it either reduces translation of that message, so that reduces the amount of protein, or it targets that messenger RNA for degradation, which also reduces the amount of protein because there's no messenger RNA to be translated in the protein. So these microRNAs, the way I like to describe them, they play a role in many, many, probably all cellular processes, but they're kind of like a rheostat. It's not on or off of the microRNA, but they can suppress levels of a protein or expression of a protein, but they usually target not just one messenger RNA, but one microRNA can target 100 or 200 different messenger RNAs. So they modulate pathways or networks that suppress that pathway, probably not blocking it completely, but modulating it. So I kind of view them as rheostats to modulate function. And we know that extracellular vesicles have small RNAs in them. They have microRNAs, they have some other RNAs, but it seems like that was the most obvious therapeutic, or the most obvious biologic for conferring this effect of stem cell extracellular vesicles. So that's where we look first. It could have been lipids. Vesicles have lipids, they have proteins, they have metabolites, they have messenger RNA at some low level, they have tRNAs. But we thought what we know about microRNAs and what's been published about microRNAs and vesicles, they were the most likely candidate for conferring the effects that we had seen with these stem cell derived vesicles.
One little detail. So it sounds like the microRNA is always suppressing a gene or a protein, but it could be suppressing a suppressing gene so that it could
Absolutely. So you do see things going up. So if you treat cells with introduce microRNAs into a cell, you see some things going up, a lot of things going down. And that's because you could be suppressing the suppressor and affecting pathways that way. So yes, although I think they function mostly by suppressing, that in turn can lead to activation of other genes. So it gets complicated, but yes, it's you do see that, but it's usually an indirect effect of the microRNA.
Okay. So which microRNAs did you find that were making the difference, and how many of them, and kind of what pathways were they accessing?
Right. So it's a great question, and it's an ongoing question in the laboratory. So what we did, and it was probably maybe too simplistic, but we took extracellular vesicles from multiple types of stem cells. And I should point out that it's only vesicles in our hands, it's only vesicles from young, healthy stem cells that do this, that have this ability to rescue senescence and rescue stem cell function. If you do this from fibroblasts or endothelial cells or epithelial cells or immune cells, we don't see it. It's not to say that there may not be vesicles from other sources that have activities. Everything we're looking at is from young, healthy stem cells. So
just so not young, healthy skin cells, only stem cells.
At least for the activities that we've seen, those vesicles don't rescue senescence. They don't rescue function of dysfunctional stem cells. And those are the assays we had used, which makes sense. Stem cells have certain properties that make them different than other types of cells. And one of them is that they can repair tissue and they can rejuvenate tissue. So we took stem cell vesicles from multiple types of stem cells, identified the microRNA content in them. We also took vesicles from non-stem cells and identified, and a lot of this was not our analysis, but it was already published analysis. And then we tried to identify the microRNAs which were in the functional stem cells, which were not in other types of vesicles. So we had a list of 15 or 20 microRNAs that came out of that were shared between functional stem cells and were not found in non-stem cell derived vesicles. And then we took these microRNAs and started to transfect each individual microRNA into dysfunctional stem cells or into senescent cells to see if they could rescue that. And what we found is that there was not one microRNA that was able to do everything. And this is one of the intriguing observations, is that a senescent cell, as we just talked about, Richard, it exits the cell cycle, it doesn't replicate, but it starts to secrete this inflammatory SASP. And we showed that these microRNAs could target different components of a senescent cell, but not all of them. So you see a couple SAS factors may be affected, but not all. We would see some markers that inhibit cell cycle progression, inhibit cells from dividing. Some of those would be affected by a microRNA, but not all. So we transfected many of these microRNAs, and we ended up coming up with a cocktail of 4 microRNAs that when you put the 4 together, suppress senescence in cell culture and suppressed it in vivo in mice. Now, these are not the only 4. There's probably many others. As we're doing more analysis, we realize that there may be better combinations. The ratio of these might need to be altered. You may want to deliver some daily, some others you might want to deliver weekly or monthly or every 6 months, depending on their mechanism and their target. So it's going to be very complicated. So we're trying to take an exosome, which has this activity, identify the components, which appears there's multiple microRNAs involved, find the right ratios, and then find a way to deliver them to mice that mimic the effect of the parent exosome. And so we've identified microRNAs, but there are probably many others. The combinations are going to be very important, and then how we deliver them back in vivo is going to be very important too.
When you're looking at the exosomes and you're trying to figure out which microRNAs are in there, is it like a binary? It's like we have this microRNA and this one, or do you know how much of each one you have?
Right. So, so if you think about a vesicle, they're very small. So that's not like the one vesicle has hundreds and hundreds of microRNAs in it. So that's one of the questions the field has is there even enough microRNA in these vesicles to confer the effects that everybody's seeing? I think the answer is yes, but it's surprising because there's not a lot of microRNA in each one of these vesicles. And so if you look at any one vesicle, probably it doesn't have all 20 or 30 of these functional microRNAs in it. So this vesicle may have some, this vesicle may have some, but if they're actually being internalized by the same cell, then that is what leads to this suppressive effect. So the way our naive view was we were going to try to find the microRNAs at the highest level, thinking they would be the most functional, and then work our way down. So we found the top 20 based on their uniqueness, but also on their abundance. And I actually think probably it's only the top 10 that may really be functional in the context of an exosome, just because there's not enough of it there to be functional. What we do is we'll make the microRNAs in the laboratory. We can introduce them in the cells at much higher quantities than what a vesicle could introduce into a cell. And then we see some activities, but I don't necessarily think they're active in the context of the vesicle because their levels are way too low. So that's a long answer to say we really pick our top 10, 15, 20 based on the uniqueness to stem cells, but also their abundance in those stem cells.
It's kind of going back to a point that I was talking about a little bit before, which is that it's only the vesicles from the stem cells that have this effect.
Right.
So there are some people who are extracting all the vesicles from, taking whole blood, removing everything, just getting the extracellular vesicles. But I think they need a very large dose. So what you're saying would imply that the reason they need such a large dose is because most of those are not functional. It's only the ones that originated in stem cells that actually help.
Well, I'm not saying there could be other vesicles from other types of cells that have rejuvenating properties. We haven't identified those yet. So what we've seen that works in cell culture are vesicles from stem cells. Um,
Right.
and so the context of what's in young plasma in regard to vesicles, I mean, very heterogeneous. What's the composition of stem cell derived vesicles in your plasma? It's got to be a very, very small fraction. But we do know, and we've done these experiments, and others have reported them, that if you isolate the bulk vesicles from young plasma of humans or from mice, that they are able to rescue, not as efficiently as pure stem cell EVs, but they can reduce senescence and appear to, excuse me, rescue function of dysfunctional stem cells. So there is something there, but it's probably a very small fraction of those vesicles. And if you look at what's in immune or what's in the circulation in regard to vesicles, many of them are from immune populations, macrophages, B cells, and then from endothelial cells, probably other types. And what we don't know is what small fraction, what is the important fraction for this rejuvenating property of young plasma. And plus, young plasma has vesicles that have rejuvenating properties, but they're also small soluble factors. There have been a number of proteins isolated which have been suggested to also have rejuvenating properties. So when you look at plasma, both vesicles and what else is there, there's probably multiple components that are found in young plasma that are not found in older plasma or plasma from older individuals. So many labs are trying to sort through what's in plasma when it comes to vesicles, when it comes to microRNAs, when it comes to proteins, when it comes to lipids, when it comes to metabolites. What are the key factors that are allowing for this observed rejuvenation seen by either transfusion of young plasma or by these heterochronic parabiosis models in mice when you pair the circulation of an old mouse with a young mouse and see rejuvenation. The other thing we're seeing, just to mention, if you take plasma from old mice and presumably from old humans, that you see that this plasma and the vesicles from this plasma can kind of increase senescence. So there actually are bad factors that you can see, which are probably contributing to driving aging. And some of these are probably bad factors or vesicles or things in vesicles. So vesicles are good when you're young, but they're probably doing not as many good things when you're older.
Yes. Okay. So you identified these microRNAs, this cocktail of microRNAs, which is having this xenomorphic or suppression of senescence. Do you know what they're doing within the cells? What pathways?
Yeah, I think that's, it gets much more difficult when you start to try to dissect out what are the key targets. Because these microRNAs, if you do bioinformatic analysis, you can identify hundreds of putative target sequences for these each microRNA. And so which of those are the important ones for conferring the effects? Which ones are just, just kind of bystander effects of these microRNAs, but don't contribute to rejuvenation. And so those experiments get very, very complicated. So we see dramatic effects on the cells because they're showing signs of rejuvenation. They're starting to look younger. But is that because we're targeting things involved in mitochondrial function? Are we changing the epigenome? Are we changing expression of cell cycle genes that will now allow cells to replicate? Are we improving DNA repair? And you kind of see changes of all these different pathways, but what's the important one? And how do we kind of improve upon that to make this effect better for trying to apply this to try and improve health span in humans? So we really don't know the key targets for the microRNAs. We see many things improving, including reduction in senescence, improving stem cell function, reduced inflammation, changes in epigenome, improving the mitochondrial function, probably improvements in proteostasis. But what's the driver? What are the key targets? We still are trying to sort that out. Because again, microRNAs don't target one gene or one pathway, they target multiple pathways. And especially if you're using the cocktail that has at least 4 microRNAs that target multiple pathways, it gets very, very complicated.
I can
Now,
see
having
that.
said all that, we've identified a microRNA that's not necessarily present in vesicles that functions as a senolytic. So if you add it to senescent cells, it actually will kill them. And this microRNA is a microRNA that was identified as being higher in human centenarians. These are individuals that live to at least 100. So these microRNAs were identified as being preferentially higher in centenarians than in non-centenarian cohorts. And so we were looking at the activity of this. So we put it through our senescence assay, and it actually functions as a senolytic. So I think microRNAs probably do many things, target many hallmarks of aging, including senescence, to help keep us healthier as we age. There's also a paper that came out recently in Cell Metabolism where they identified a microRNA in vesicles. It's a specific microRNA, different than the ones we isolated, but they have said that it extends health span and lifespan in mice when delivered that one microRNA. So they were calling it a rejuvenating microRNA. And so, there may be many of these that have positive effects on aging, not just the 4 we've identified, but a senolytic, this rejuvenating one that was identified in other types of stem cells. So I think there's a lot still to learn about what microRNAs are the right ones to work with and their targets.
Are you working on that at the moment, like trying to find out whether there's a bit better cocktail?
Yes, absolutely. And we're taking multiple approaches. The first approach is looking to see what we can find in stem cells that are functional as compared to vesicles that are derived from non-functional stem cells or from non-stem cells themselves. And so that's where we identified this cocktail. But there are other factors there, other microRNAs that we're characterizing. The other approach we've taken is we've actually screened for microRNAs that are upregulated or downregulated in senescent cells. So we've developed a reporter system where we can screen for microRNAs that are functionally upregulated with senescence or functionally downregulated. So we've identified a number of microRNAs that if you introduce them in the senescent cells, suppress senescence, and microRNAs, if you put them in the normal cells, drive cells into senescence. And those microRNAs, many of them actually are known tumor suppressor microRNAs. So these are microRNAs that have been identified genetically as being mutated in human cancers, because they were thought to be tumor suppressors. And so our model is that some of these microRNAs are actually tumor suppressor microRNAs because they drive cells with oncogenic mutations into a state of senescence, which is how senescence evolved. But we've also identified microRNAs which will drive cells out of senescence. So they're ones that potentially could be useful for aging. And then the last thing we're doing, so as you may be aware, there are companies like Altos Labs. They're trying to do partial reprogramming, so allowing rejuvenation of aged cells. So this is the process that was identified by Dr. Yamanaka, won the Nobel Prize, for showing that you can drive non-stem cells into an embryonic stem cell-like state, so that you can actually reprogram non-stem cells to become stem cells. But if you only do that, start that programming process a little bit, cells look younger and healthier. And the question is, how do you apply this to humans in a safe manner? Because there's a cancer risk with some of the reprogramming methods. So we are also looking for microRNAs that are induced during this partial reprogramming. And we've identified those that go up with reprogramming and those that go down. And we're now testing those to see if they can rejuvenate cells like the other partial reprogramming methodologies. And the answer appears to be maybe. as we do, but we have more experiments to do. So we have multiple microRNAs that can suppress senescence, can improve stem cell function, and actually can rejuvenate non-senescent cells, but age cells. These are usually, we're using dermal fibroblasts, but can actually rejuvenate them so their transcriptional profile looks younger, more like a dermal fibroblast in the younger individual. So those are the approaches we're taking, but now we have basically a large number of microRNAs, and you have to sort through them to determine which one or which combination of those is going to be the most effective. And that's kind
Is
of
there,
where we currently are. Very excited, but a lot more work to do.
yeah, I can certainly see that it is really complex.
Yes.
So, is there any specificity between the type of stem cell that the exosome comes from and the tissue that it works on? I mean, so yeah.
Yeah, it's a great question. And I think the answer has to be yes. Um, but we're still scratching the surface on that. So if you have a stem cell present in adipose tissue or present in muscle or present in liver, are they making vesicles that go everywhere and target all cells, or do they target specific cell types? And I think the answer is going to be that they don't go everywhere, that they're preferential cells, which these vesicles bind to and internalize into. We just don't know um, what the key targets are, both just what's internalizing the vesicles, and then functionally which cells are being improved by stem cell derived vesicles. And it's an important question because we want to use the right vesicles to target the right cells that then confer this effect on Healthspan. And if we could identify what the right targets are and improve that targeting, that also would lead to better therapies.
Do you see, have you done much in vivo? And do you see like the mice becoming epigenetically younger?
So we have not done very much in the context of looking at the epigenetic clock. We're now starting to, but there have been other reports, not from our group, but others who've also injected stem cell derived, I think in their case was adipose stem cell derived vesicles, and showed that in certain tissues, they could see the epigenetic clock looking younger. What was intriguing was if they stopped treatment, so they were giving vesicles every week or two, if they stopped treatment, the clock reverted to an older phenotype. So this suggests that the rejuvenation is transient, at least at the epigenetic level, and I think also at the transcriptome level. So you probably have to give vesicles or these microRNA cocktails at a certain frequency. Is it every week? Is it every month? Is it every 6 months? In humans, well, we don't know. But I do think there's going to be rejuvenation at the epigenetic level, at the transcriptome level, and other hallmarks of aging because they're all linked.
Have you thought about how this could be translated? I mean, is it possible to manufacture a microRNA and deliver that, or would you use exosomes, or would you try and create a drug?
Well, I think at this point, again, the billion-dollar question. At this point, the vesicle has evolved to really be the best delivery mechanism. And if you have the right stem cell source, it has the right microRNAs and seems to confer these beneficial effects. The problem with using stem cells or stem cell-derived vesicles is every time we isolate primary stem cells from a mouse or a human, expand them, and when you expand them, that decreases the stem cell function of those stem cells, which leads to vesicles that are not as active. So when you isolate them, expand them, and isolate the vesicles, there's huge differences between preparations. So what you need is a stem cell line that can be expanded, 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 that doesn't change its function, and the quality of the vesicle stays the same. And then methods to purify enough vesicles, because we're injecting into a mouse uh, roughly 10 to the ninth particles. And we think we have to give 1,000-fold more to humans. So we're talking 10 to the 12 vesicles, that if you have to give them every couple of weeks for years, it's a lot of vesicles. So this is very expensive. And just the quality control, we see huge differences because we don't have a stem cell line that has all the right properties, at least not yet. And so when we do this with primary stem cells, each prep has different activities just because of the variation. The age of the person or age of the animal, how they were isolated, how they were expanded, isolated the vesicles. So the alternative is to try to come up with the right microRNA cocktail and the right lipid particle that is effective at targeting the right cell types, just like the vesicle, the extracellular vesicle, is targeting. So I think that's where we will eventually end up because it's going to be controlled in a much better manner. The cost of production is going to be much less, I think, than stem cell vesicles. However, I think right now, the most effective thing we have are stem cell vesicles. So I think some of the first trials that are going to, there are already clinical trials using stem cell vesicles for other disease indications. They're just not being given systemically for aging. So I think those experiments will start. They're going to be expensive. There's going to be, there are going to be issues about producing enough to give frequently to an aging human. But where we'll end up is eventually a microRNA cocktail in a lipid particle that can be targeted to the right cell types. And then the other big question is how many cells in the body do you have to rejuvenate? So, if you rejuvenate 10%, do they provide a beneficial effect to their neighboring cells? If you rejuvenate a liver, and there's evidence that if you make a liver healthier in the diseased animal, that mouse lives longer and healthier if you improve kidney function. So, making some cells in the body rejuvenated might have systemic effects. So you don't have to rejuvenate all cells. Rejuvenate some, and those cells then in turn rejuvenate others and rejuvenate others. So don't know this, but these are in questions if you have to do partial reprogramming of all cells, that's going to be difficult. But if you only have to kind of improve the youthfulness of 10% of your cells to see effect on Healthspan, that may be more attainable, or 1%. So these are the other questions we have to address, is really how widespread do you have to have this stem cell EV distributed in the body to see rejuvenation and an effect on Healthspan.
Right. And you'd think you preferentially would like to rejuvenate stem cells.
So if you could rejuvenate stem cells in vivo, which we don't know if we're targeting in vivo or not, but if you could, then that would in turn, those stem cells would be able to improve other tissues, and that effect spreads. So yes, that would be one goal, and it could probably be done directly by the vesicles going into those stem cells, or by reducing inflammation, improving the stem cell niche, as we talked about earlier. There may be many different ways of improving stem cell function in vivo.
Yes, but stem cells generally are mutagenic. Oh, not mutagenic. They will be rejected, right? If they're from a non-matching donor.
Which is a problem with using allogeneic stem cells. The question about whether extracellular vesicles from allogeneic stem cells are rejected at the same rate. And the answer is clearly not. But will they eventually stimulate the immune response so that those vesicles will lose their effectiveness after multiple dosings? And there's data that suggests that might be the case, and there's data that suggests that's not the case. Because some people have been able to treat mice with vesicles derived from an allogeneic source, so not matched. They have different proteins that are going to be antigenic, and they can inject them daily for months and still see biological activity. So it may depend on the source of stem cells. It may depend on route of administration, may depend on the purity of the vesicles. There may be a lot of factors that contribute to that, which may alleviate some of the immunogenicity concerns you see with allogeneic stem cells. due to the fact these vesicles are not as antigenic or immune stimulatory. And many of these vesicles appear to be suppressive. They can suppress immune function. So, because they reduce inflammation, they reduce chemokine expression, maybe because they're targeting senescent cells.
Right. Are you aware of the work of Dr. Katcher using? So he used young pigs, exosomes from young pigs' blood in rats,
Right. Right.
and that well, at least it wasn't. It didn't promote an allergic reaction because the rats were fine even after being injected. So it would seem to imply that exosomes are not allergenic.
But there are reports of people doing multiple injections of vesicles and losing the activity of those vesicles after multiple injections. Now, why that activity is lost, is it due to an immune response, an antibody response to the vesicles? It's not clear, but there are reports of loss of activity, which could be due to induction of immune response. So it may depend on the types of vesicles, the route of administration, the dose, the purity. There may be a lot of factors. So it's really hard to compare between experiments. But the feeling is that vesicles are going to be less immunostimulatory than cells. And so therefore may be safer. And they can't transform. So stem cells can acquire mutations. They can, potentially themselves lead to cancer. They can become transformed themselves in theory, but vesicles can't. Vesicles don't have DNA. They don't proliferate. They don't change their phenotype if you put them into a host. So the thought is they're going to be much safer than some of the cell-based therapies, and at least hypothetically less immunostimulatory.
The microRNAs that you've identified in your cocktail, are they well conserved? I mean, would they be the same microRNAs in mice and rats and pigs?
So far, the microRNAs that we identified in human vesicles, they work in mice. So there is some conservation on their target sequences. Now, is the mechanism going to be identical? They may have some different targets between mice and humans, but at least microRNAs identified in human stem cell vesicles have shown effects on mouse cells and in mice after delivery. So I think the answer is yes.
Well, it it does seem very interesting. I mean, the having got this, having identified like these particular RNAs and just the beneficial effects. But as always, it always seems to get more complicated when you get into the details.
Biology is complicated, and aging is very complicated too. So if you put just biology in general together with aging, I mean, this is not a genetic disease where we know the specific mutation that drives beta thalassemia or muscular dystrophy or cystic fibrosis, which have been very difficult to treat, despite the fact we know that the protein which is mutated. Aging, everything goes wrong. So, it's much more complicated. But if we can target aging, then all, not genetic diseases, but most age-related diseases, the severity will be reduced or alleviated completely if we can actually slow aging. So I think the approaches we're taking to target aging, even though we don't understand all the mechanisms, it will have the largest impact on human health of anything that's being worked on today. So this concept of targeting aging as a way to treat age-related diseases, I think makes perfect sense, and hopefully will be changing medical care in the future.
Yes. So, is there anything else that you think would be good to talk about in this area that I didn't cover, that we haven't covered?
I think we've covered a lot. I think it's the importance of using stem cell based approaches, or using what we think are the key factors for conferring the effects of adult stem cells. that this obviously is an area that needs to be explored in much further detail. And I think it's going to have to take clinical trials to be initiated to really push this field forward. I mean, it's going to take, yeah, if we can show that we see positive effects in humans, if it's slowing aging or if it's improving frailty or if it's even treating, more temporary conditions, this will move the field forward much, much faster. People are, worried about the expense. They're worried about just safety issues. But I think a clinical study showing improvement in the context of an age-related disease or aging itself will move the field forward very, very quickly. And so I'm hopeful that this happens. We're probably not going to be the ones to do it, being an academic lab, but I think some of the companies in this area are focused on this, and I think there will be data coming, hopefully in the next 3 to 5 years, that will show, yes, this works, or no, this doesn't in humans.
So the cocktail that you're currently using would be, I mean, would be something that could be triable in a trial.
And if you were to ask me how we would do this, I would say the first target would be skin aging because of safety and probably ease of delivery, and also cost because you could do it in a very localized area. So I think delivery to the aging skin and then measuring markers, which you can do through biopsies of the skin, this will be a first step to showing, yes, this potentially could work if given systemically. But that would be my guess of maybe the first next step when it comes to clinical studies.
Is your work with these microRNAs published? Yeah.
We have a paper just accepted in Aging Cell, a paper just accepted in eBiomedicine, and 2 more submitted. So they will be published. Some of them will be published shortly, and then hopefully the others will be following quickly behind those. But we and others, there are a lot of people working in this field. So in addition to our papers, there are others coming too. So I think in the next year, you're going to see a lot of microRNA studies for aging and microRNA studies of, the therapeutic content and extracellular vesicles. So there's going to be, so I think this field hopefully will become even more exciting over the next year or two.
I shall look forward to that, and I shall watch for them. So if people want to follow your work, where can they go?
It's a great question. We're talking about putting on the newsletter. We haven't done that yet. My website keeps an up-to-date list of papers that are accepted or been published, which can be found at robbins.umn.edu. So UMN for University of Minnesota.edu. So my website tries to stay current. And we're going to try to have more of a social media presence. We're meeting later this week to talk about how to be disseminating all the results from our Institute on the Biology of Aging and Metabolism, how to disseminate that to the community, to get the word out about this whole concept of geroscience, and that things are coming which might help us stay healthier for longer. So there will be better ways to follow my work in the near future. But right now, I think my website's the best way. Hopefully the newsletter, social media presence, even talking about doing a podcast similar to what you're doing, Richard, that we can help share some of this information. So I really appreciate this opportunity. I always enjoy talking with you. You're very insightful, very knowledgeable. So it's always a joy to have these discussions.
Thank you, Dr. Robbins. It is always great to talk to you. And yeah, thank you so much for your time.
All right. Have a good day. Have a good